Welcome to Season 2 Purdue University College of Sciences Superheroes of Science podcast. I'm Stephen.
And I'm Sarah. We will be discussing anything and everything related to science. If you have a science question, tweet it to us at Purdue SOS, and we will try and find someone to answer it for you. Joining us today on Superheroes of Science, we have David Thompson, Professor of Chemistry at Purdue University. So welcome, David.
Hi, how are you?
Doing
Good.
well.
Yeah.
So Sarah, Stephen,
Yes.
it's I've not done this before. I'm really curious to see what I can share about my.
Well, let's start. What types of research does your group do? What types of research do you do?
We are organic chemists at the core of the laboratory capabilities. So what that means is we like to make and break bonds to make new compounds. Almost always, sometimes there's a metal in there. We often use metals to make our compounds, but they're usually just composed of the same things that we are: carbon, hydrogen, nitrogen, oxygen. They're organic molecules that we believe will have interesting properties that we are hoping to design into those molecules. So in some projects, those molecules are designed to be a drug. In other words, a pharmaceutical kind of compound. In other cases, those molecules are designed to actually do a better job of delivering the drug. So that if, let's say, simple comparison, a regular aspirin and what's called enteric coating aspirin, something that releases the aspirin over a longer time period. That's a drug delivery kind of application. Same active drug, just different way, different that it impacts the human body. So those are two big areas of our research activity. We are also involved in making new materials that we think will be useful for determining structures of enzymes, viruses, something that's in the news these days. Many people have probably seen some of the cartoons of the coronavirus. They're actually, many of them that I've seen they're not really cartoons. They're kind of low resolution structures that come from electron microscopy. And that's the area that we are trying to impact without getting too deep into the weeds. There are many challenges in determining determining some of those structures. Often a challenge is actually making a decent sample. You may have a coronavirus sample that you want to look at in high resolution. Pardon me. But you can't do it because the sample was not prepared correctly. So strategies to do a better job of making the sample is another area where we're active in making new materials.
How does someone go through doing that?
Pardon me?
What's the process? How would you go through preparing a sample of a virus to be able, as you say, to look at? And how exactly are you looking at this virus?
Yeah, okay. Yeah, so I'll try to come back at that question in a second. I want to, in a way, kind of finish framing the idea that very much what we do as chemists is it's the same thought process as is often used in engineering, where you design it, you build it, you test it, you then go back to the drawing board and you redesign and make something better. So there's this design, test, analyze, redesign. And that's very much what we do with organic compounds as well, whether it's a new drug that we're trying to develop or a new drug delivery strategy, or in the case you're asking about, preparing samples for virus analysis. We make a material and then we test to see, can we see the structure of the virus in higher resolution or not? So specifically, we're looking in the electron microscope. One of the explosions that has been happening, and I mean that in a good way, one of the, I'll let me change the word, revolutions that is happening in structural biology really began in the mid 80s. And the people who kind of built the foundation for that, they were awarded the Nobel Prize in chemistry 3 years ago. Roger Higginson, Duche, and memory test. I can see his face. Frank. For developing a technique. And basically, what it does is you flash freeze a very thin sample of your, let's just say, virus. And now you need to get that tiny, very thin slab of ice into the electron microscope, which is at a super high vacuum, and shoot electrons through it. And how those electrons interact with the sample gives you information about how to deduce the structure. And I guess this is the only prop I have available is my phone. So if this is the object, the beam passes through the sample in this direction. You see the projected image looks like a rectangle. But if the beam goes through the sample in this direction, it's now the projected image is a different kind of rectangle. And likewise, as the sample is in all different orientations, you get a lot of different projections. You can take those projections and essentially reassemble. You can do a so-called reconstruction of that object, that three-dimensional object. And that's actually what you're seeing when you see those coronavirus images are usually reconstructions of microgroups of electron microscope images where that technique has been used. Now, if the ice is too thick or the virus was damaged in the freezing process or enzymatically degraded, there can be a million ways that it goes wrong. You end up with a bad sample. And so you have to start over. So that's part of what we're trying to do is put into place a more reliable way to make those samples. Here's maybe a useful piece of information. The typical of photograph, or at least single particle images that need to be taken to do a good reconstruction, it can be as many as I've seen publications where they took 4 million different particles in different orientations. And now they have to figure out all of the particles that were in that orientation, add all those up, take all the ones in this orientation, add all those particles up, and in that way kind of build up the signal to noise, build up the reliability of the information. That's a lot of work. And so how do you, and so made a bad sample, oftentimes you don't know that until you've gone through all the work of isolating it, freezing it, getting it into the electron microscope, and then capturing images. And then you look at the images and you go, oh, it's like back in the day. Now I'm dating myself. Maybe some of your instructors know this. Everyone used to have film cameras and take a bunch of good pictures, and then it would come back and it's like, oh my God, they're all terrible.
Yep.
Yeah, somebody was scratching their nose when the shutter went off, and so gosh, you have to start over. This you'd like to have a more efficient process. That's what is really kind of the heart of
Oh, very nice.
particular material design.
So what seeing that
Um,
help?
pardon me?
What does getting a good image of something like a virus, how does that help us what does it help us understand?
Oh, wow, excellent question. So the goal is to get an atomically precise picture. Because if you know, if we stick with the virus example, if you know a particular interaction, here's a cell surface, the cell about to be infected, and the virus particle binds to a certain cell surface molecule that allows that virus to be internalized. If you know exactly the shape, it's like knowing the the structure of the inside of the lock. You know exactly the key to cut that will fit in that lock. And in this case, it's kind of in that way, it's a bad analogy. You'd like to block that infection by having something bind more strongly than it will bind to the cell. If you can do that, now that virus can never get inside the cell because you've blocked it with an inhibitor, with a drug that will interfere with the normal infection cycle of the virus, or the microbe, or the enzyme that's misbehaving in a disease process. So that's what the electron microscope is trying to do, is to get precise enough images for us to do a better job of designing drugs to improve human health.
Okay, so didn't you work on different compounds that would like fit that lock, to use that analogy?
Yeah.
Part of what you researched then, just obtaining the picture and determining, kind of generically put, the shape of these things?
That is where we are trying to get to. And we're trying to develop methods to allow us to get there. There are, you can more or less count on your two hands the number of structures that have achieved that kind of precision that will allow for design, for you to really precisely cut the key.
Mm-hmm.
But the ambition is for us to develop the science and make it robust enough that it will actually allow for medicinal chemists to be more precise. Because the way it has been done and is still done today is through a lot of just testing. You isolate if you want to inhibit an enzyme or whatever. What's a what's a good case? Alcohol dehydrogenase. If you want to inhibit that enzyme for some reason, you know the structure, you know the molecule you have to design. These days, if you don't know the structure, you are often taking libraries of tens of thousands, or if you're a pharma company like Eli Lilly, you have libraries of as as many as 4 million compounds. And it's like the locksmith in the old days, if you were locked out of your house, they came with a big key ring that had hundreds of keys, and they're there testing the one that gets you in through the door. And that's more or less the way a lot of drug discovery still happens today, is from that just massive scale. So the more you can get knowledge about what's the strike zone, what's the actual size and shape of the target you're trying to hit. Now you can design, you going back to that lock and key. I'm kind of mixing metaphors here, but go back to that lock and now do a much better job of designing the key.
There's two terms that we've used, and I kind of like to find so that to make sure, especially our younger audience, understands the terminology we're using. One was you talked about creating compounds, and the other was using enzymes. And so,
Yeah, okay.
What exactly are these two things?
Great question. So compounds that's a very generic term, and a compound can actually be any composition. It can be any collection of atoms. Most often, especially in the area where I'm active, it's usually referring to a relatively small molecule. Relatively meaning it is smaller than an enzyme. An enzyme is typically as many as it can be thousands of amino acids that are all strung together in a chain. And so they are, that would be a very, very heavy compound compared to most drugs, which are, have the, actually, if you look at something like the physician's desk reference, more than, which is essentially a book of all the commercially available drugs. Over 90% of those drugs are way less than 700 hydrogen atoms. To put that in context, enzymes, thousands of amino acids, those are going to be as many as hundreds of thousands of hydrogen atoms. Give you maybe that gives you some bookends to think
Mm-hmm.
about it.
Mm-hmm.
An enzyme, though, to be precise, an enzyme is essentially it's a catalyst. It's something that will make a reaction go faster. And the body is filled with them. Uh, we as chemists try to make compounds or uh, catalysts that are they're not made from amino acids like enzymes are, but we try to make catalysts that are, if you can make one that's even half as good as a bad enzyme, you're a hero. Enzymes are the champs. That's what we're trying. We'd like to be that good, and that's what we work hard to try to mimic in a artificial, with artificial compound, or I should say, non-natural compounds.
So you talked a little bit about with your students and all students really when they go about trying that process of testing and experimenting. And so can you describe the process that a student might take with thinking of an experiment that they want to try and then how they would go from there?
Yeah, yeah. So it usually starts with a hypothesis. We expect that, let's say, I'll take a drug delivery example, that we can vary the rate at which a drug becomes available in the body varying part of the structure of our drug delivery system. So it starts with that hypothesis. And we'll use an example. I'll use specific example. Let's say we want the drug to become available in the stomach. The stomach has a lot of digestive enzymes. It's actually low pH. And so you think, oh, maybe we'll use low pH as a way to activate or release the drug. Or maybe there's a specific enzyme that we can use to make the drug become released by the drug carrier. And so that's the core starting point. Then, as chemists, we go in and we design a compound. If it's pH, if we think it's going to be activated at low pH, then we think, oh, okay, this reaction, the more the lower the pH, this reaction goes faster. That's a good reaction for us to build around. So then we draw a structure, then we go to the lab, we make it, and then once we've made it and proven to ourselves that it's been made, then we actually do the test. So that's the part of this design, build, test cycle. Design, that's the idea, that's the picture you draw on the whiteboard when you're having the you and the student are having a conversation. You're doing that design piece. To build it, that's actually going into the lab and actually, as I started, making and breaking bonds to build the new maybe conceptually no different than getting a bunch of dimensional lumber and taking the plans for your house and all that lumber and you fit it together to build a house. You know, it's kind of the same process. But then, you know, maybe you realize, oh, I put the spacing too far apart on my in my roof, and I got a leaky roof, and I now I have to go back and redo that part of my design. You know, that's this design, build, test, uh, analyze, and redesign. And so, in essence, the process and the uh, part of what makes it interesting is to figure out where uh, the right strategies to get you the answer that you're trying to answer the question that you've raised or the hypothesis that you're trying to test. Part of the way you design your testing strategy is to get to that answer as quickly as possible because you know time is the most limited element and that's precious resource. That's what you want to try to answer those questions as quickly as possible. And actually, quite often, that is linked to how much time you've spent really thinking carefully about the question. Have you posed the question in a way that will give a yes or no answer, or will give a very clear readout, a very clear answer during the analysis? And that's where a lot of the creativity and kind of planning a sequence of experiments that will get you as quickly as possible that answer. Because it is, after all, it's not only time, it's competitive, right? We're
Right.
competing with all the other scientists around the globe. And you don't, it's just like in the newspaper business, the one uh, one entity gets the lead story, right? Everyone else is kind of following. It's not being second isn't nearly as exciting as being first.
Yeah. Well, David, thank you.
We really appreciate your time today, going through, first of all, the what you're researching and what your students are doing, and then also that whole design and test and redesign process. That's very helpful. So thank you.
Yeah, yeah.
Definitely.
Well, it's hopefully the listeners can really take that to heart. And one other comment is to because that whole set of experiments you're trying to put in place, you always need to do controls. What that means is there's a so-called positive control, which is a test where you already know the answer, and the answer is yes. So you build that into your experiment so that when you do that test, that tells you if you see a yes answer, now you know your test is working. And if you get a no answer, you know your test is bad. So that's what's called a positive control. And then you also have to have the inverse. You have to have a negative control, an experiment where you know that the answer is no. And if you do the test and it comes out yes, again, it tells you there's something wrong with your design. And so those are really key. Those controls are key, and it's really important to do those first because I'm going to give you two perspectives on this. One is the time perspective that I was just talking about. If you, let's say, in the positive sense, you get a yes answer from your test, and you did your real experiment, the one that is going to really answer whether your design is good, your new molecule is good, and you get a yes answer, you get all excited and you do the next experiment, and finally you do the control and you realize that all of it was what's called a false positive. It was actually something wrong with the way you were measuring. And actually, all along, you were uh, collecting useless data. That feels really bad.
It's
I
like a
bet.
lie detector when you calibrate a lie detector for your research.
Exactly. Or in today's world and coronavirus testing, where the news is that even the very best tests are only 90% accurate, right? So that 1 in 10 people out there may have a reading that says they're negative and they're actually positive. Um, so it's crucial, actually. That's a way to kind of really bring it home to you to what's current.
Mhm.
But I think the temptation when you're so excited, because if you're a scientist, you at this, when you're at this level, you're really driven to explore new phenomena. And when you have led yourself down a garden path like that, and you realize all the money and time you've wasted, that's a lesson you never forget. But it's even better if you're a young scientist and learn to always do the controls first.
That's a nice way of getting back to that. Very nice.
Yes.
Well, thank you again. We
Yes,
appreciate you here.
thank
We
you.
really appreciate you taking the time for this. And
Okay.
Thank you for listening to our podcast.
Be sure to subscribe, rate, and leave a review
An outstanding
on
review.
iTunes or your preferred podcast player.
Tweet us your science
At
questions.
Purdue SOS.
Until next time, be super and remember,
You are someone's hero.
boiler up.
Hammer down.